my name is Reagan McRae, and today I will be presenting my group's research study entitled Familial Taxonomic Determination of Rodents from Humoral Measurements, conducted by myself, Maddie Kinneary, Emma Catlett, Makenzi Pryor, and Stacey Clark, along with our faculty advisor, Dr. Timothy Campbell. For our research, we led with the question, can rodent humeri be used to identify specimens at the familial level? We chose to look at rodents because they are prolific in their global distribution and exhibit a wide range of diversity. The humerus was chosen to look at for a few reasons. First, it is a four-limb bone, meaning it would have different jobs within the body based on whether the specimen is a biped or a quadruped. Second, it articulates with multiple other bones, such as the scapula, the radius, and the ulna, which again could cause differences due to how the forelimbs are being used. Finally, the humerus is subject to environmental and locomotive pressures, such as arboreal locomotion, as compared to terrestrial locomotion. In order to test our question, we used a collection of 267 photos of humerized specimens from the southern African region that belongs to 8 different families in order to collect measurement data. Image J was used to collect 4 measurements from each bone: the maximum length of the humerus, the minimum diameter at the neck of the humerus, the epicondylar breadth, and the maximum diameter at the deltopectoral process of the humerus. That raw data was then entered into Excel to be size adjusted through natural log and geometric mean calculations. The size adjusted data was then put into PASS version 4.04 and run through a principal components analysis for data reduction and visualization. The PC scores 1 through 3 collectively account for 86.9% of the variance retained for further research. The scores were then entered into a multivariate MANOVA test with the families to find the p-value and the Wilks lambda score. The p-value was less than 0.001, and the Wilks lambda was 0.16, both highly statistically significant scores. This confirms the statistical significance between the humeri measurements and their families. In conclusion, the answer to our initial question is yes. Rodent humeri can be used to identify specimens at the family level. However, it is important that further research is done in this area. First, a larger sample size with more taxonomic diversity would be helpful in showcasing a wider range of specimens in order to determine differences. Geographic diversity and familial diversity will both help in this area. It would also be beneficial to take more measurements per specimen for data processing. Finally, taking the data from the study and running it through a discriminant function analysis will help determine whether the differences found in the study are actually usable differences to be used for future studies. Thank you so much for listening. Before I close, I would like to thank everyone that helped our group to succeed in this project. I would like to thank our faculty advisor, Dr. Timothy Campbell from Baylor University's Department of Anthropology, for providing us access to the data and photographs, as well as helping us along this process. I would also like to thank all 5 collections that allowed us access to their files: the American Museum of Natural History, the Smithsonian National Museum of Natural History, the Field Museum of Natural History, the Natural Science Research Laboratory Museum of Texas Tech University, and the Ditsong National Museum of Natural History. And finally, I would like to thank the entire Department of Anthropology at Baylor University for granting us the opportunity to conduct our research. This project would not have happened without all of the help and support we have received. Thank you.